Hello, I'm Dr. Chris Baldessano, and I'm an assistant professor in psychology at Columbia University. Today I'll be talking about recent work in my lab to understand how the brain's response to a narrative movie can differ within an individual as we learn about the movie, and can differ across children of different ages. To start, let's review some of the key features in neural responses to narratives that I and others have studied over the past few years. One characteristic feature that we see in the brain responses to narratives is that there are time periods during which there are stable event patterns. Here's one way of looking at the data that can show this type of event pattern. What we have here is a time port correlation matrix. We're taking pairs of time points from different times in the movie and looking at the spatial pattern correlation between the brain's patterns during those two time points. If there's a stable event pattern for some period of time, that means that all of the event patterns during some time period are all going to be highly correlated with one another, and then there'll be a jump to a new pattern. In this example, we can see this big block of high correlation here. These are all pairs of time points that all have a high spatial pattern correlation. And then we can see at this moment here, we suddenly see a switch. Now we have a new set of time points that are no longer correlated with the old set of time points. This kind of division into events, into stable patterns of events, is something that we see in responses to movies and narratives, anytime that the brain is doing some kind of event segmentation process. A second characteristic feature is that the brain maintains multiple timescales of events. So in addition to these relatively large chunks that can take seconds or minutes, we also see these smaller kind of event chunks, during which brain patterns remain stable just for a few seconds at a time. If we look across the brain, we can see regions, especially sensory regions, that have these relatively short event durations, during which these periods of stability are relatively short. And then in some of these higher order regions, like these posterior medial regions and regions stretching out into the ventral visual stream, we start seeing these longer event patterns. We start seeing patterns of activity that remain stable for longer periods of time. And we think this kind of hierarchy is really important for maintaining a representation of what's going on in the movie, for capturing not just what's currently on the screen, but also what's been going on over the past few minutes, and building up these situation models of complex events. Finally, multiple studies have shown that narratives tend to evoke strong hippocampal responses at event boundaries. Here's an example from Ivan Yakov that shows that at these event boundaries, we get a large change in the hippocampal response. This is just measuring univariate activity across the whole hippocampus and shows that when we hit an event boundary, we see a big response, and that response scales with the strength of the boundary. So if there's a high salience boundary in the movie, meaning that there's a very big change between two events, we get this large hippocampal response that is triggered by this event boundary. So in this talk, I'd like to describe some recent work in my lab looking at how these kinds of narrative movie responses can change. First, we'll be looking at what happens with repeated viewing of a movie. Once we have knowledge of what's coming up in the future, how does that change these kinds of movie responses? And then we'll talk about how these responses change across development in kids ages 5 to 19.
How can you read that again?
It's good.
Yeah, but you've read it like 1,000 times.
I enjoy it.
I always thought the pleasure of a book was in wanting to know what happens next.
When we rewatch a movie that we've seen before, it gives us a special power. We're able to anticipate upcoming events in the movie by using our memory. We use this kind of anticipation all the time in our daily lives, and it's one of the main reasons for having an episodic memory in the first place, since it helps us to make decisions or prepare specific actions ahead of time.
He has special powers.
Yes.
He can see things before they happen. That's why he appears to have such quick reflexes. It's a Jedi trait.
But how does anticipation work in a continuous stimulus like a narrative movie? There isn't one specific outcome to predict. We could try to anticipate at short time scales, for example, anticipating people or objects that are about to come on screen, or we can anticipate at longer time scales, thinking about plot twists that are minutes away. One hypothesis comes out of the literature on how we represent the past. Previous work has shown that during perception, we're tracking information at multiple timescales. Some brain regions have very little memory and react only to the current stimulus, while others accumulate information over seconds or minutes, and then use this context to help understand and interpret what is happening in the current moment. What we don't know is whether there's also a hierarchy of anticipation for future upcoming events. When watching a familiar movie, does the brain maintain representations of upcoming events at multiple timescales simultaneously? A recent project in my lab sought to answer this question. This project was led by Carolyn Lee and is a collaboration with Dr. Miriam Ali. In this project, we took data from a previous paper, Learning Naturalistic Temporal Structure in the Posterior Medial Network. This paper included a few different types of fMRI datasets, but the piece we're interested in is what they called the intact movie. This is simply a 90-second movie. This is a clip taken from the Grand Budapest Hotel, and each subject watched this movie 6 times. There were other stimuli too, so they weren't 6 back-to-back times, but it was all during a single experimental session that the subject was watching this movie repeatedly. To give you a sense for what this is like, I'm going to play the movie twice in a row. The first time through will be the first time you're seeing this, unless you've seen this movie before. And then on the second time, you'll be able to predict what's coming up next based on your memory of the first repetition.
Thank you, Mr. Mosher.
You're most welcome, Monsieur Gustave.
You're now going to be officially interviewed.
Should I go and light the candle first, sir?
What?
The
No. Experience.
Hotel Kinski, kitchen boy, 6 months. Hotel Berlitz, mop and broom boy, 3 months. Before that, I was a skillet scrubber.
Experience 0.
Thank you again, Monsieur Gustave.
Straighten that cap, Anatole. Pleasure's mine, Herr Schneider.
The stress busters.
These are not
I
acceptable.
fully agree.
Education.
I studied reading and spelling. I started my primary school. I almost finished.
Education 0.
Now it's exploded.
Good morning, Cicero. Call the goddamn plumber.
This afternoon, Monsieur Gustave.
Will that fail, Frau Liebling?
What on earth is this?
Not now. Family.
0.
6, Igor. Why do you want to be a lobby boy?
Well, who wouldn't? At the Grand Budapest, sir. It's an institution.
Very good. Thank you, Mr. Mosher.
You're most welcome, Monsieur Gustave.
You're now going to be officially interviewed.
Should I go and light the candle first, sir?
What?
The
No.
candle.
Experience.
Hotel Kinski, kitchen boy, 6 months. Hotel Berlitz, mop and broom boy, 3 months. Before that, I was a skillet scrubber.
Experience 0.
Thank you again, Monsieur Gustave.
Straighten that cap, Anatole. Pleasure's mine, Herr Schneider.
The stress busters.
These are not
I
acceptable.
fully agree.
Education.
I studied reading and spelling. I started my primary school. I almost finished.
Education 0.
Now it's exploded.
Good morning, Cicero. Call the goddamn plumber.
This afternoon, Monsieur Gustave.
Will that fail, Frau Liebling?
What on earth is this?
Not now. Family.
0.
6, Igor. Why do you want to be a lobby boy?
Well, who wouldn't? At the Grand Budapest, sir. It's an institution.
Very good.
To look for anticipation in this dataset, we're going to fit a model called a hidden Markov model. This model makes a few assumptions about what fMRI data looks like in response to a movie. First, it assumes that on every repetition of the movie, we're going to see the same sequence of event patterns in a given region. So whether it's the first time or the last time watching, your brain is going to proceed through the same sequence of spatial patterns. But across repetitions, the timing of these patterns might differ. So which pattern is currently activated at a particular time might be different across repetitions. The goal of using the hidden Markov model is to try to learn this sequence of event patterns, so to understand what these spatial patterns are that are coming in this sequence during viewing, and to try to assign every time point of each repetitions to belong to one of these patterns. This is a probabilistic model, so the assignment we're going to get is going to be a soft probabilistic assignment. At every moment in time, we're going to get a probability distribution over what event we might be in at that moment in time. Here's a toy example of what this looks like. We're going to take the data from some brain region as our input. So here you can see that we have this brain data of voxels and time, and we have this data for each of the 6 viewings. So the goal of the HMM is to take this data. There's no additional labels provided to the HMM besides just these 6 matrices. And the goal of the HMM is to produce 2 things. First, it's trying to figure out what the discrete events are that underlie the activity in these brain regions. And it's going to come up, in this case, we're looking for 4 events. It's coming up with a sequence of 4 event patterns that the brain is passing through on every viewing. The other thing the HMM is trying to learn is this assignment of time points to events. It's trying to figure out for every time point, which of these 4 events do we think that this time point belongs to. So here you can see these matrices. These are not voxel by time matrices. These are now time by event matrices. So you can see that in the first viewing, we think that we start in the first event and we stay there for about halfway through the stimulus. Whereas in this last event, the model is estimating that we move out of this first event and into the second event after only a couple time points. One way of summarizing these kinds of matrices, these probability distribution matrices, is to just take the expected value of the event at every moment in time. What that means is we're basically estimating on average which event do we think we're in at every moment in time. That allows us to produce plots that look like this. And so what this is showing is over time for each of the 6 viewings, which event are we in at each moment in time. You can see that the first viewing, which is the yellow line, is the lowest on this plot. That means that it's proceeding through the events the most slowly. The darker lines, which are the later viewings, are higher up on this plot. That means that they're transitioning into these later events earlier on in the stimulus. So for example, here, this is about a third of the way into the stimulus. The first viewing is still stuck in event 1, whereas these later viewings have already progressed to event 2. Now this is just toy data, but this is the kind of analysis we're going to be applying. We're going to do this in a searchlight across the brain. We're going to be looking for regions where there's this big separation between the first viewing, when people don't know what's coming up next, and these later viewings, when people can use their memory to anticipate what's coming up next and potentially transition earlier into these later events. Here's what these results look like across the cortical surface. Every colored in region on the cortex here, it shows regions in which these later viewings are significantly ahead of the first viewing, that there's a significant amount of anticipation on these later viewings. We're coloring these regions. The different colors indicate how much anticipation there is. So in these later viewings, how ahead are they in seconds compared to this first viewing? How much earlier are we transitioning into these events relative to the first time that we showed the movie? And so what we can see here is that, first of all, we can see anticipation in many parts of the brain. And we can also see that this anticipation varies across the brain. If we look in a relatively low-level sensory region, like here in lateral occipital cortex, we can see that there's only a small amount of anticipation. Here on the bottom, we're plotting this expected value plot. We're showing which event the HMM estimates this region is in during each of the viewings. And you can see that the lines are quite close together. There are a few moments in time when the later viewings are a little bit ahead of the first viewing, but in general, there's not a very large separation. We can then move up the cortical hierarchy. Here we're going into supramarginal gyrus. And here we start to see more separation between the viewings. So we can see that on later viewings, they are on average, about 6 seconds ahead of the earliest viewing. We're transitioning into these events. We're moving through this sequence of event patterns more quickly on the later viewings. And finally, if we look at some very anterior regions in the brain, we can see relatively large separations. So here we're seeing an anticipation of almost 13 seconds for these later viewings compared to the first viewing. That what we see is that we're transitioning into these later event patterns in the sequence much earlier on in the stimulus. You might wonder how this approach compares to a simpler option of cross-correlating the time series. We could take the time series of Voxel activation in response to the first viewing of the movie and compare that to the time series of Voxel activation to the later viewings of the movie. The idea is that we could correlate these two time series, and we could also cross-correlate them with different shifts. We could take this later viewing time series, and we can move it forward and backward. The goal will be to identify what is the lag that provides the best match between the first viewing and later viewings. We would identify this maximum lag, this lag that maximizes the correlation, as the amount of anticipation. If there is a region that is anticipating on later viewings, we'd expect that we would need to move this viewing's 2 to 6 time series forward in time in order to match these viewing 1 time series. If we try to use this approach, we do get some significant results, at least in some of these more anterior regions in the brain, where we're able to identify a simple shift between the time series that is happening with repeated viewing. However, this approach is relatively insensitive. One assumption that this approach makes is that there's a constant shift that's happening throughout the entire movie. The HMM approach is able to be much more flexible. We can identify shifts in timing that vary across the movie. Maybe there are only particular moments when we can actually predict into the future, or the amount of prediction varies a lot depending on which part of an event that we're in. Using the simpler approach, we are not able to identify this full hierarchy. We're really just getting the largest effects near the top of the hierarchy, and we're not able to see all of these different amounts of anticipation. This is one reason why we think that this modeling approach is much more effective for identifying anticipation as compared to using this simple cross-correlation approach on the time series. One question we had is how this hierarchy relates to the intrinsic timescales in these regions. As I mentioned before, we think that each of these regions has some kind of timescale at which they accumulate information during perception, and we were interested in whether this timescale is related to the amount of anticipation. So in order to figure out what the intrinsic timescale of each region was, we took half of our subjects, labeled them group 1, and then we fit the HMM using just these group of training subjects. We in fact fit multiple HMMs with different numbers of events. So for example, we fit an HMM with just 2 events to this group, we fit an HMM with 3 events, an HMM with 4 events, and so on. In order to figure out which of these models was the best, we then took each of these models and tested how well they explained the brain activations in a new group of participants. So we took a second group of subjects, an independent group of subjects, and tested how well these models explained their activity. The idea here is that we can use this to identify the intrinsic timescale of a region. That when we've set the number of HMM events correctly for a given brain region, then we should get good transfer between these two groups. If we set the number of events too low or too high, then we're not going to do a good job of explaining the group 2 data based on our fit to group 1. So we can run this kind of analysis, this kind of cross-validation analysis in every region in the brain and see what this intrinsic timescale looks like. So here are the results I was showing before, the anticipation results. And here's what the map looks like of these intrinsic timescales. And so what we can see is that these also vary across the cortex. We see the fastest intrinsic timescales. These are regions with the most number of events around sensory regions, especially around auditory cortex. In other regions that are colored in yellow, these are regions in which a small number of events was the best fit to that particular region's time courses. And we can see that these two maps are related to each other. So for example, if we take our anticipation results on the left and bin them based on the results on the right, we can see that there's a relationship between these two quantities. So the x-axis here is showing the different bins of optimal event timescales. This is the duration of events that was the best fit for a given brain region. And then we can, on the y-axis here, we're showing what the distribution is of anticipations among regions with that intrinsic timescale. And so what we can see is that these longest anticipations tend to be in regions that have these relatively slow event timescales. So regions that accumulate information over a long period of time are also the ones that are anticipating farther into the future. Regions that have a very fast intrinsic timescale, so they're changing their patterns very quickly, they're representing things that are just currently in the moment, we don't see a lot of anticipation with repeated viewing in these areas. I should mention that these fits of optimal event timescale, these are being fit just on the first viewing of the movie, and then we're comparing them to this amount of anticipation that we get in the later viewings. Another question we had is how this anticipation relates to what's actually going on in the stimulus. So the HMM itself doesn't know anything about the movie or the content of the movie, but what we wanted to figure out is whether this anticipation means that the brain is actually ahead of the stimulus, meaning that these changes in event patterns actually precede the change that's happening in the movie, or is it the case that we're just getting more responsive to event changes in the movie? So the first time we see the movie, when there's a scene change, so for example here, there's a scene before getting in the elevator to getting in the elevator. Maybe the first time you see this movie, your brain is a little bit slow on the uptake. It takes a few seconds to transition into this new event. And then on later viewings, you're much faster. So for example, we could see that on this first viewing, you stay in one event, meaning that your brain has some spatial pattern of activity. We cross over the boundary, and then a few seconds later, you start building up a new situation model. Later on, we see that we're much more responsive, that when this boundary happens, our brain is able to switch more quickly. And so this would show up as anticipation in our analysis. However, it's also possible that our brain actually starts responding before this boundary happens. Since we know that this scene change is coming up, we could start representing that new situation even before it happens on the screen. So we wanted to run another analysis to try to distinguish between these two possibilities. Are we just getting more responsive to boundaries? That is, we change more quickly after a boundary happens, or are we actually anticipating in advance of the change happening in the stimulus? So to do this, we first had to collect some ratings of where event changes are in the stimulus. That's shown in this histogram here. These are all of the annotations that our raters made of where these boundaries are in the stimulus. And the height of these bars shows the number of raters that rated each of these as a boundary. Then what we did is we took these boundaries, this histogram of boundaries, and convolved it with a hemodynamic response function. This is generating a hypothesis about when we would expect to see these boundaries show up in the brain. So you can see in the stimulus, there are 3 large scene boundaries, as well as some smaller ones that were rated by a smaller number of people. So now we have our behavioral measure of where humans think these boundaries are in the movie. And what we'd like to do is compare that to our brain data. We have these model fits from the HMM showing these transitions between events. And so we'd like to get a kind of boundary measure out of this expected value plot. And we can do that by simply taking the derivative of this plot. So just looking at the changes in expected value at every moment in time. So if the model is transitioning between events, then we'll see a large value of this event change, meaning that the probability distribution is shifting a lot during this moment in time. Moments in time when the model thinks that we're not switching between events, we'll have a low value of this event change. So then we can take these hand annotated boundaries and correlate them with these event changes. We're going to correlate these 2 different time courses at different lags with each other to try to figure out what the relative timing is between a boundary happening in the stimulus and it being reflected in these event patterns in the brain. We can first look at this region in the middle temporal gyrus. What the plot is showing here is on the x-axis, we're showing the lag in the cross correlation between the brain measure and our stimulus measure. Each of the lines here shows the correlations for one of the 6 viewings. So what we can see here is that the first viewing, which is the yellow line, starts out relatively well aligned with the stimulus. If we look at the 95% confidence interval of this first viewing, we find that it includes 0, meaning that the correlation between the brain and the stimulus, these are relatively well aligned on the first viewing. However, in later viewings, we see this shift, that now the brain is actually transitioning its event patterns before there's any change in the stimulus. And the shift is somewhere in the range of 2 to 4 seconds in these later viewings. So what we see here is that this anticipation is actually bringing the brain responses earlier than anything that's happening in the stimulus. that we're starting to see transitions in the brain before we've actually had a transition in the stimulus. We've also found significant effects in some other regions. In the fusiform gyrus, we saw something similar, though here on the first viewing, we're actually a little bit behind what's going on in the movie. So the brain's neural response is maybe about 1 or 2 seconds behind the stimulus response on the first viewing, and then this shifts to be about a second ahead of the stimulus on later viewings. If we look in the superior temporal gyrus, we see another pattern, which is that the brain is actually quite far behind the stimulus on the first viewing, so maybe up to 8 seconds behind on the first viewing. And then on later viewings, we've now shifted to being ahead of the stimulus. These later viewings are at least a couple seconds ahead of the stimulus transition when we see these neural event patterns. So to sum up this study, we found that event responses shifted earlier in time with repeated viewing, and that the magnitude of this shift increased across the cortical hierarchy. We saw small shifts in lower level sensory regions and larger shifts in these higher level regions. We also found that these event transitions in the brain came to precede boundaries in the stimulus in some brain regions. So this anticipation was actually happening before there was actually an event boundary in the stimulus itself. We have code to reproduce all these analyses that's available on our GitHub as well, if you're interested in trying this on your own data.
This will all make sense when I am older. Someday I will see that this makes sense. One day when I'm old and wise, I'll think back and realize that these were all completely normal events.
In the first part of this talk, we used a design in which people were shown exactly what was going to happen. But even when we see a movie for the first time, we can make use of our general knowledge about the world, and perhaps knowledge about the kinds of narrative arcs shown in movies, in order to structure our perception and memory of the stimulus. How do narrative responses change as we accumulate more experiences with age? This project was led by Dr. Samantha Cohen. What Samantha did was took a subset of data collected by the Child Mind Institute. She used 415 subjects with ages that range from 5 to 19. All of these subjects watched the same 10-minute movie. I'm going to play a clip from that movie now.
And Agnes, clink. Very good. Excuse me, girls.
Oh, come on.
Don't worry, I'll be back. Keep clinking. Oh, Miss Addie, what are you doing here?
I'm here for the girls. I received a call that you wanted to return them.
The first analysis we ran was to look where in the brain there are changes in the response to this narrative movie. What we wanted to do is compare the time course in the youngest subjects, the 5 to 8-year-olds, with the oldest subjects, the 16 to 19-year-olds, and identify regions that showed different average responses across the age groups. For this particular analysis, we weren't interested in the variability within the age groups. We wanted to ask whether the mean response, the group average response, was different in the two groups. So our idea was to compare correlations within the groups versus across the groups. The idea is that if there's some kind of shift in response with age, if there's some kind of systematic difference in how the younger and older children respond to this movie, then we'd expect there to be more similarity within the group and less similarity across the age groups. What we did is constructed this young-old similarity correlation index. On the numerator, we're looking at the correlation between the two groups, between the young and old subjects. And then in the denominator, we're taking the geometric mean of the correlation in the young group and the correlation in the old group. If there's no systematic differences between the groups, if the groups have the same mean response, then we would expect this ratio to come out to be about 1. We'd expect the similarity across the groups and within the groups to be about the same. If there is some kind of age-related difference, then we'd expect this index to come out as less than 1. We'd expect the correlation across groups to be less than the correlation within the groups. So what we can do is divide the brain up into parcels and compute this young-old similarity index for every one of those parcels. All of the colored parcels here have a similarity which is significantly less than 1. We can see that in some of these regions, the similarity is pretty close to 1, so that indicates that there's only a relatively small shift in age. However, in some of these regions, we see a relatively large shift. These regions include the temporal parietal junction, TPJ, the dorsolateral prefrontal cortex, DLPFC, the inferior temporal cortex, and orbital frontal cortex. In these regions, we see a correlation between the groups, which is as low as 0.6. So this indicates that there's a change in the average response of the older kids and the younger kids in response to this narrative movie. In follow-up analyses, we tried to identify what specifically is changing in these regions. And so one hypothesis is that these changes have something to do with the event structure that's happening in response to the movie. So remember, we can use this hidden Markov model to assess this event structure, to try to explain the responses in a region as the brain moving through a sequence of event patterns. By applying the HMM, we can measure changes, first of all, in the extent to which this event-structured hypothesis fits the data in a brain region. So we can measure the log likelihood of the model, meaning we can measure the extent to which a brain region looks like it has an event-structured response, where the brain sits in one event pattern for a little while, and then rapidly switches to a new event pattern and remains stable in that new pattern until it switches again. We can also use the HMM to assess the timing of event transitions, just like we did in the first half of this talk. We can look at whether these two things change with age. Do we see changes in how event structured the response is across age? And do we see changes in when these event transitions happen, when there's switches between events? First, we'll look at whether the changes in the log likelihood of the model, the extent to which this event-structured model fits the data. First, let's look here in the left TPJ. What's being plotted here along the x-axis are models with different numbers of events. So at the leftmost side, we have a relatively large number of short events. At the rightmost side, we have just a couple of very long events. We're plotting different lines here for each age group. And so you can see we're fitting these models of different sizes, different event sizes for each of these age groups. What we see in this region is that, first of all, the model fits tend to be better in the older subjects. So in the darker lines, this event-structured model tends to be a better fit. We can see that for these older subjects, the best model fit is somewhere around the 1 minute scale. What this is telling us is that only in these older subjects do we get this very event-structured patterns in the left TPJ at this 1 minute time scale. So in the older subjects, we're seeing them switch into an event pattern and remain stable for up to a minute. And in the younger subjects, we don't see that kind of stability in this region. Looking across the brain, we see a few different regions in which this is true. So we see the later part of auditory cortex, as well as in visual cortex. We see this better event structure in these older subjects, as well as in parts of the frontal lobe, like in DLPFC. There are a couple regions where we actually see better fits that are in the younger groups. So for example, in this posterior medial region that's indicated in this plot here, we see better event structure in the younger groups. This could be due to a number of reasons. It could be that there's more diversity among different subjects here, that as the children are getting older, there's a wider range of different responses in this region. It could also be that there's less stability across time, that in the younger subjects, we see fewer dynamics in these regions. We see more stability, and in the older children, we see more dynamics. We can also use the HMM to assess the timing of these transitions. And we can look in each brain region to see, does it look like older children are transitioning sooner than younger children? Unlike our previous experiment, none of the children were shown this movie during this experimental session. It's possible some of them have seen it before, but it wasn't in the recent past. So what we're interested in is whether there's something happening with age here, where older children are able to better anticipate upcoming events in the movie. And so we can see that this is true in a number of regions. This is true in left TPJ, left DLPFC, and left precuneus. These plots are showing the transition timings relative to the youngest group. So we can see that in some of these regions, like in left precuneus, we see that the oldest group can be up to 20 seconds ahead of the youngest group in terms of when they're transitioning between event patterns. Again, in this posterior medial region, we see a reverse effect, where the younger children are actually a little bit ahead of the oldest children. This is a relatively small effect, but it could indicate that these younger children are more responsive to event boundaries. They're switching more quickly when there's an event, and these older children are lagging a little bit behind. They're still thinking about the previous scene. That's only true in these particular regions here, these blue-colored regions. In general, we see that the older children are anticipating events, that their event transitions are happening earlier than in the younger children. Finally, we can also ask about what happens in the hippocampus at boundaries. So remember, I mentioned in the introduction that we usually see a hippocampal boundary response anytime we see an event boundary in a movie. Previous work looking at an aging population has shown that this hippocampal boundary response decreases with age. Here we're looking at 3 groups of subjects: a young adult group, a middle-aged adult group, and an older adult group. We can see that this boundary response is present in all of the age groups, but is substantially lower in the oldest group. They also found that there were differences in different parts of the hippocampus. This plot is showing the posterior hippocampus, and here's what it looks like in the anterior hippocampus. So what they found is that this change with age is primarily happening in the posterior part of the hippocampus. The question in our study is, do we see changes in hippocampal boundary responses over this younger age group, between ages 5 and 19? And do we see a difference in different parts of the hippocampus that's driving this change? First, we just looked for every participant, we looked across the entire hippocampus at what the boundary response was. What we found is this robust hippocampal event boundary response in all of our age groups. And we actually saw a small decrease across this age group. What this meant is that the youngest children, the 5 to 8-year-olds, actually had the largest hippocampal responses, and then this decreased with age. If we break up the hippocampus into anterior and posterior portions, we actually found that this effect was being driven by the anterior hippocampus. So we found that the anterior campus showed this significant decrease with age, whereas this change was not significant in the posterior hippocampus. So what this tells us is that there is this event boundary response. It's present even in the youngest children, and in fact, it's largest in these children, especially in the anterior part of the hippocampus. This was a surprising result to us. We didn't expect to see this larger response in the younger children. One possible explanation of this is to think about potential trade-offs between the hippocampus and the cortex. We think about building these situation models and building memories as a kind of collaboration between pieces of the hippocampus and parts of the cortex, with the cortex dealing with more of these generalized schemas, and then the hippocampus binding the particular details of this particular episode. It may be that younger children rely more on their hippocampus, and especially their anterior hippocampus, because they've yet to build up these strong schemas in the cortex. It could be that over time there's a trade-off between using the hippocampus to bind all of these details versus offloading some of this into the cortex as we're able to build up better schemas. Further work will need to be done to figure out how this kind of trade-off works. To summarize this part of the presentation, we found age-related changes in narrative movie responses in a number of regions, including TPJ, DLPFC, PMC, and OFC. We found that in several of these regions, including TPJ and DLPFC, the responses became more event-structured with age, meaning there was more pattern stability within events. And we also saw this anticipation, that event transitions shifted earlier with age. Finally, we found that children in all age groups showed a hippocampal boundary response at event boundaries, but that this response decreased in the anterior hippocampus across this age group. Again, code is available to replicate all of these analyses if you're interested in applying them to your own dataset. In ongoing work, we're trying to understand how responses to a movie can change not just with age, but can also depend on the experiences of an individual child. In collaboration with Dr. Nim Tottenham, we're showing children one of 2 different movies. Both of these movies involve the separation of 2 characters and then the later reunion of these characters at the end of the clip.
We've fought this kind of battle before, haven't we? And you never cried once when I went away. Remember?
Yes, Daddy.
Well, this is going to be our hardest battle. But we'll be good soldiers, won't we?
Yes, Daddy.
not such good soldiers as we thought.
Oh, yes, we are. I can do it now. My daddy has to go away. But he'll return most any day. Daddy?
Sarah.
Daddy! Oh, Daddy, it is you. I found you. I found you. They said you were dead, but I knew you weren't. I knew you'd come back. Oh, Daddy. Oh,
Yes. Yes. Sarah. Sarah! My baby.
oh Daddy, you
Oh, my
do
darling.
know me. I'm gonna miss you so much.
I know. I know you're sad. I wish I knew why.
We'll be back to visit next week, okay? I promise.
What's wrong?
It was old. It was too far. It was just too old. Oh, Peter, honey.
Peter.
Shadow!
Oh, Peter. I worried about you so.
What we're interested in seeing is how the responses to this movie might be different, first of all, in younger versus older children, but also in children that may have formed different kinds of schemas about attachment. Part of the sample of that's used in this experiment are children that have faced early childhood disruptions in caregiving, and we might expect them to have formed different schemas and different expectations about what's going to happen in these clips. I've looked at this question of schematic responses in the brain in previous work. For example, I presented participants with different narratives that conform to either a restaurant or airport schema, and I found that there were brain regions that responded similarly to all of the stories within a schema. Our hypothesis is that children will show this same kind of schematic response, but that these schematic responses will be disrupted in children that have not been able to form traditional kinds of attachment schemas. I also wanted to mention some exciting work being led by Tristan Yates in Nick Turk-Brown's lab at Yale. They've been measuring the fMRI responses to narrative movies in infants less than 1 year old. They've been playing these same narrative movies to adults for comparison. So for example, here's what the adult response looks like to one of these clips. Here again, I'm showing one of these time point correlation plots. It's showing what the spatial pattern correlation is between pairs of time points in the movie. And in adults, we see these blocks along the diagonal, indicating that there's moments in time when the patterns are relatively stable. If we look at infants, we actually see a similar kind of structure. We see that the infants also have these periods of pattern stability, during which the pattern in a given brain region stays relatively constant before it jumps to a new pattern. There are some differences from the adults. For example, the infant events tend to be coarser. They tend to be longer in duration than the adult events. The infant events are interesting in other ways too. We actually see that these infant events that are picked up from the brain often correspond to the annotated events from the stimulus. So this gives us some idea that the infants are actually constructing event representations that are relevant to the stimulus in some way, and changes in the stimulus are leading to changes in the pattern that's being represented in infant brains. This is really exciting line of work, and there's lots of other questions that we can ask here about what infants are doing and how this changes in these very young children. I'd like to thank my lab members, my collaborators, and my funding sources for all of their support, and to thank you for listening. Links to all of the research I discussed in today's presentation can be found on our website, dpmlab.org, with links to preprints, as well as reprints of all of our published papers. You can also find out more about the lab members that conducted this research by clicking on the People tab.